Hi, Brian here and welcome to the Biology Tribe, a show where we discuss everything about biology and medical sciences including molecular biology, biotechnology, microbiology, and biochemistry. In today's video, we are going to discuss the Central Dogma. This video covers the history of the Central Dogma, the definition of the Central Dogma, an explanation of the concept of the Central Dogma, the steps involved in the Central Dogma, and the exceptions of the Central Dogma. This video comes with additional 2 steps in the Central Dogma and unpacks the most current information about the exceptions of the Central Dogma. Stay tuned till the end of the video. Let's get into it together. Starting with the history of the Central Dogma. Francis Crick, the discoverer of the DNA structure, first developed and proposed the Central Dogma theory in 1958. The Central Dogma of molecular biology states that genetic information flows only in one direction, from DNA to RNA, to protein, or RNA directly to protein. Let's begin with defining the Central Dogma. The Central Dogma refers to the process in which the genetic information flows from DNA to RNA to make a functional product, which is the protein. Now let's explain the Central Dogma briefly. In molecular biology, Central Dogma illustrates the flow of genetic information from DNA to RNA to protein. The Central Dogma shows the generic flow of genetic information in cells, DNA replication, and coding for the RNA through the transcription process and other RNA codes for the proteins by translation. According to the Central Dogma, DNA has the instructional data required for making all proteins. The RNA serves as the messenger for carrying these instructions to the ribosomes. Then the ribosomes act as the industrial units within the cell that help translate the instructions from a code into an easily understandable product called the protein. The conversion of DNA information into readable products is called gene expression. Biological frameworks such as biopolymers can help better understand the conceptual sequence of interaction in the Central Dogma. Biopolymers include DNA, RNA, and proteins, which are grouped into special, unknown, and general transfers. While the special transfers occur under exceptional cases in the lab, the general transfers happen in nearly every cell and involve the movement of information via transcription and translation processes. Information on whether the unknown transfers occur remains unclear. The 4 steps involved in the Central Dogma. The major stages of the Central Dogma are transcription, splicing, translation, and replication. The first step in the Central Dogma process is transcription. During transcription, the polymerase enzyme and other transcription factors guide the transfer of information in the DNA strand to the RNA. The concerned DNA molecule has 3 sections: the terminator, promoter, and structural gene. The DNA strand that makes the new RNA is referred to as the template strand, while the other is called the coding strand. The DNA-dependent RNA polymerase attaches to the promoter to catalyze the polymerization in the 3' to 5 direction. When it nears the terminator sequence, it terminates and produces a newly synthesized RNA strand that undergoes post-transcriptional enhancements. The second step is splicing. Most articles don't discuss this step, though it is also worth mentioning. Splicing occurs in eukaryotes, and it involves the processing of the primary transcript, that is the pre-mRNA. One or more sequences, that is, introns, are cut out. The mechanism of alternative splicing makes it possible to produce different mature mRNA molecules, depending on what sequences are treated as introns and what remain as exons. The next step is translation. Most scholarly works indicate this as the second most crucial stage in the Central Dogma. In the translation stage, the RNA codes the information from the DNA template into specific proteins. The translation is an active process that requires the energy provided by charge transfer RNA, put as tRNA molecules. The translation is initiated by ribosomes, which can be either larger or smaller subunits. The larger ones, in turn, have 2 tRNA molecules positioned so close that they can form a peptide bond as soon as there is sufficient energy. The mRNA proceeds into the smaller subunit, which is then held by the tRNA molecules of the complementary codon within the larger ribosome subunit. Therefore, the two tRNA molecules closely positioned holds the two codons, and a peptide bond is formed between them. The process repeats itself, and longer polypeptide chains of amino acids are formed and begin folding into the correct conformation. The folding continues until the nascent polypeptide chain is attained from the ribosome as a mature protein. In some instances, the newly forming polypeptide chains need further processing to make a mature protein. The proper folding process is a bit complex and may need additional proteins called chaperone proteins. Sometimes, the proteins splice themselves further, and when this occurs, the inside disregarded part is called intine. The last step of the central dogma is replication. To transfer the genetic information down the lineage, the DNA has to replicate correctly. During replication, a group of complex proteins unwind the superhelix and unwind the double-stranded DNA helix. Moreover, DNA polymerase and its related proteins imitate the master template to ensure the cycle recurs from DNA to RNA to protein in a new generation of organisms or cells. However, the central dogma does not hold at all times. This brings us to the exceptions of the central dogma, which contradicts Crick's conceptual approach that genetic information flows only in one direction, from DNA to RNA, to protein, or RNA directly to protein. Crick postulated that genetic information does not flow from proteins to nucleic acids, and modern discoveries have established a number of these exceptions. Some species of viruses are so primitive that they use only RNA for protein processes without developing into the DNA. One particular exception is that of the prions. During infection, prions replicate without going through DNA or RNA intermediates as they involve protein-to-protein steps. This means that proteins directly replicate themselves by making conformational changes in other proteins. Another landmark revolution in central dogma study is the discoveries made in retroviruses. Retroviruses transcribe RNA into DNA, a process catalyzed by reverse transcriptase. In the case of retroviruses, their central dogma follows transitions from RNA to DNA to RNA to proteins. Even though certain primitive viruses, prions, and retroviruses have exhibited these exceptions to the central dogma, the rule that all cellular life forms adhere to the central dogma still holds because these groups of organisms are not considered as being alive. Current research focuses on investigating the function of non-coding RNA. Although this does not follow the central dogma, it still has a functional role in the cell. Thank you for watching. And if you found this video helpful, please give me a like, subscribe, and turn on the notification bell so that you don't miss being notified when I upload new content. And let me know your thoughts in the comments section below.